I came to Providence Cancer Center back in 2001 to work with Andy Weinberg and Walter Earle on the OX40 immunotherapy project. Now, as a basic scientist, I never actually thought I'd get a chance to work on something that would actually be put into humans. But it's been a lot of fun. It was a little scary at times. But I'd like to take this opportunity to thank Andy and Walter for giving me that opportunity. Now, I found out when I got here that my part of this big project was in fact entirely funded by the Portland community, you guys. And Walter and the Medical Foundation raised about $1.5 million or something to bring OX40 therapy to patients. So I would just like to thank you for making this opportunity possible. So as you have heard, immunotherapy is in the news today. There's a lot of attention being given to immunotherapy. And you may have, you just heard it, and you may have seen Emperor of All Maladies, where the last half hour was devoted to immunotherapy because they saved the best for last. It's on the cover of Science magazine, which you can't see right now. It's the journal Science. So I'd like to say that the attention is deserved, right? We are getting cures in advanced cancers that were never possible before. However, it only works in a minority of patients, in a minority of cancers, and we need to, and we're working hard to do better. Now, but back in 2001, many in the medical and scientific community never thought this could possibly work. So, in a very real way, you and we together were pioneers in cancer immunotherapy. So, I'd like to give you my version of what immunotherapy is like. So, what we know is that the immune cells can kill cancer cells. That immune cells go to the cancer, they spew a bunch of toxic molecules at the cancer cell and kill it, and then they move on to the next cell and kill it too. And, but the rest of the body has to be protected from this onslaught. So immune cells have a whole series of on and off switches, typically proteins on their surface, that the immune system can use to ensure that the immune cells are turned on at the right time and place and turned off when they're done with the job. Unfortunately, early in the evolution of cancer and the development of a cancer, mutations can happen, changes that allow the cancer cells to make proteins that hit the off switch. So the immune cell goes to the cancer, but instead of killing it, gets shut down. So immunotherapy, the goal of immunotherapy is essentially to try to block those off switches and also to hit on switches to try to reinvigorate the immune response. And what I left out, but want to say anyway, is that we can actually see this after surgery. If you look at the tumor, you can see the immune cells all around the outside. They're there, but they're not doing anything. And so hopefully, when we block those off switches and hit on switches, we can get those cells to start killing the cancer. So what about OX40? So OX40 is a very powerful on switch. So when it's activated, the immune cells divide, so you get more of them, and they make more of those toxic molecules that they use to kill the cancer cell. And when we try this therapy in mice, it works really well, so we're really good at curing cancer in mice. However, Andy and I needed to come up with a way to try to do this in patients. So we developed this antibody. Now, this antibody is it recognizes and binds OX40. It binds one OX40 at each end of that Y, at the tip of that Y. So when it binds OX40 on the surface of a cell, it activates that on switch. We made this antibody in mice, and we were able to get a clinical trial here where we could show that it was safe, that it activated immune cells, and it had an impact on the cancer. It could shrink the cancer somewhat. Now, this is a mouse protein, so after a week and a half of treatment, the patient's immune system blocks or rejects that foreign protein, and so we can only give one round of therapy. So we wanted to convert that mouse antibody, anti-OX40 antibody, into a human anti-OX40 antibody, and ultimately we want to get FDA approval. Now, this is a huge undertaking, well beyond the scope of what we can do here, probably in the hundreds of millions of dollars of effort. So we partnered with AstraZeneca, one of the large pharmaceutical companies, to take this project forward. What we did is we gave AstraZeneca the mouse antibody, and they converted it to a human antibody. So they replaced all the mouse parts and made human parts. And we gave them this funny looking molecule that Andy and I dreamed up in the lower left-hand corner. I won't really try to describe it to you other than to say, well, the antibody can bind 2 OX40s. This other protein can bind 6 OX40s. So it gives a much more potent signal to the immune cell to act as a much more powerful on switch. So MedImmune took the mouse antibody, the humanized antibody, and this funny protein and started a whole series of clinical trials which began last fall. So in these trials, we're going to hit the on switch with OX40, and AstraZeneca has 2 antibodies that bind off switches, CTLA-4 and PD-1, and PD-1 was the one you just heard about. So the goal there is we can hit the on switch, we can block the off switch at the same time. Think of it as stepping on the gas and taking your foot off the brake at the same time. That we think will give a much more powerful immune response, and hopefully we can extend the reach of immunotherapy beyond where it currently is today. Now, these trials are occurring, we're going to start with all comers, but essentially eventually we'll focus on the kinds of cancer listed there. Now, what we're going to try to do is ultimately to find the best combination of on and off switch with OX40 plus one of these other off switches in the most responsive cancer, so that we can try to get to phase 3, ultimately to phase 3, where we can prove to the FDA that it works. And this is, as you imagine, a huge effort. Next slide. It's a nationwide set of trials. Providence is one of the sites. And up to about 500 patients are potentially going to be treated. Brendan Kurdi and Ram Leidner are principal investigators on these trials, so Providence is shouldering a significant burden for executing these trials. Now, lest you think that OX40 is the only thing going on in immunotherapy, next slide. Here's a list on the left, the top 20 list of potential targets. These are all on and off switches. The top 2 have been approved. The rest are all in clinical trial. So the goal is to try to figure out which one of these work, in which combination, with which of the standard therapies, and have to work through what order, what doses, which cancers, which patients, because in fact, cancer today is a very individual disease. We're in the era of personalized medicine, so each patient presents a unique therapeutic problem. So OX40 is 4th on that list now, so we're not the only one that thinks it's important. We think it actually should be a little higher on the list. But we're pretty excited about where we are. Now, Providence is fully engaged in all of this process. As Walter mentioned at last year's luncheon, 4 new first-in-human trials were started, and so we're contributing to this list. Our clinicians are fully engaged in working out through these trials and trying to answer these questions. So I guess what I'm really trying to say is we still need your help. It turns out that working on cancer, on a cure for cancer, doesn't actually protect you from getting cancer. So 2 years ago, I was diagnosed with prostate cancer. I was fortunate in that it was caught early, but it was among the more aggressive types. So I had surgery, and it was a good result because there's no evidence that it had spread. But there's definitely a possibility for recurrence. So I initially, well, like perhaps many others of you, I don't know, obsessed a lot about probability, what's the chance of it coming back, how many years do I have, but as a friend pointed out to me, either it comes back or it doesn't. So instead I decided to focus on the fact that I can see what's coming. So you saw that list. There's a lot of things coming down the pipe. And so that if cancer comes back 10 years from now, 5 years from now, 1 year from now, therapy then will be better than it is today. But I also take even more comfort in the fact that here at Providence, there is deep knowledge in the science and art of immunotherapy. We've been doing it for over 20 years. So if my cancer comes back, I know that I'm already at the best possible place I could be, and in fact, I just have to walk down the hall. Thank you.